Welcome to Scientific Communication, the first video I'm making for this summer course. It is going to go over how to communicate scientific findings in the written format, so scientific writing. So we're going to go back into class where we've just done our first experiment of microbes in the environment. We've seen our results, and we made some awesome discovery. So now what do we do with that? We're going to want to communicate the results. So there are different audiences that you're going to communicate to, and there are different ways that you would present this information. So several ways to do this. We're going to start today and focus on scientific writing. Later, we'll talk about poster presentations and oral presentations and some other videos. Those are other ways that we would present this information to the scientific community or to the public. So writing is typically how we do this. So we primarily work through journal articles, which are peer-reviewed and go through a rigorous process of editing, basically. There are two different types of sources. There are primary sources and secondary sources. Primary would be where you're reporting new results. You've done firsthand something and you're going to tell people about it. Secondary sources are going to be sort of if you're summarizing a bunch of things into a new paper, think like you're introducing a topic to someone. And then there are news articles. So these are also a secondary source, but this isn't aimed at scientists or people that would be replicating your results. This is aimed at the general public. So we're going to focus on science writing today. Science writing. So it starts with your abstract. So here are all the parts of your traditional science paper. Your abstract is an elevator pitch. It's your one paragraph summary. Think about it in a few short sentences. You want to tell the reader why you did, what you were looking for, how you did it, what you found, and why it was important. But you got like 30 seconds to do so on an elevator. That's what you're writing here. So it's typically written last, but we're going to start with it because it's sort of an easy enter into this writing realm. Your introduction is your why. So that's where you start off and you tell people, why did you bother doing this? What's the background info they need to know before they read your research? Why is it important? What gap does it fill in science? What was your hypothesis? What's that question? And then your methods is your how. Think of it like a recipe or procedures what exactly did you follow and that somebody could read this section and do your experiments again. The results, this is just the what. What did you find? So you followed those and you said, I found a red ball. I found 5 millimeter long worm. I don't know, whatever you found, you're going to report directly in words and pictures your data and observations. You don't need to relate it back to that introduction yet. You don't need to tell us why this is important. You just need to tell us what you found. The discussion is where you say what those results mean. So why did they come out the way they did? Did you have errors or something like that? Or was there a variable you didn't take into account? Are they important? Does your results support your hypothesis? What does this mean in the greater scheme of the world? And what would you do next? Where are we going with this? What would you hope somebody else would take out of this? The last bit is references. This seems simple. Plagiarism is really bad. In science, plagiarism, you cannot quote things like you would in a traditional English paper. In science, you have to paraphrase everything. So there should be no quotes directly taken from a source. You must rewrite it in your own words, cite it inside the paper. Typically, you'll have lots of citations in your introduction and your discussion section. There won't typically be citations anywhere else. And then you'll have a full references section that will allow us to find those papers that you referenced. Here's your abstract. We're going to dive deeper a little bit. So your abstract is short, concise. You want to think of it like a challenge to summarize your entire experiment in as few words as possible. So this should be able to stand on its own. It could be, basically, you'll be reading papers and you'll be reading abstracts and you won't read anything else from the paper. As you're trying to find information, we don't need you to have a fully deep understanding of everything for this level of science. As you move on, you may need to read more and more, but you should be able to read the abstract and understand what the whole purpose of the paper and what they found was, whether or not it be relevant to you. If you find it to be relevant, then you might move on to the introduction to see what the basis for all of that was. So your abstract is going to cover all of these things: your context, your content, and your conclusion. So it's going to have all three things in it. So in your basically one paragraph, the one question you're answering is this: here is what we did, here is what we found, and this is why it matters. So you have your background and your research question, your results, and why it matters. I've just said it in a couple of different ways there. Then we move on to our introduction. Our introduction provides that context. It's that deeper. It's the sort of, think about that as your English paper when you talk about a topic, about writing. This is that background where you're going to say, hey, these are microbes. This is where microbes live. This is what they can do. This is why we are interested in this surface. Why we might care one way or another. So what does the reader need to know? And you would start at a pretty basic level, even though your audience is another scientific reader. You also want to make sure you start really broad. Think of this like if a 2-year-old were to be asking you questions about your project and you said, hey, yeah, I swabbed the kitchen sink and I found X number of microbes. And they're going to be like, why? Well, because there were microbes growing on the kitchen sink. Well, why? Because it has a lot of water. Why? Why do we care about microbes? Why did you do that? Why did you pick that surface? Why? So if you can keep asking yourself why until you get back to the very basic of microbes exist and what they are, and then the why we wanted to look at that surface and why it matters to us as humans to tell us what your hypothesis was. It's going to have that sort of funnel effect. Go from really broad into the really specific. This is what we did today. This section would have lots of references where you would include all of your citations to previous research that had been done that gives you that foundation for where you're going with your research. Your method section, this one tends to be one of the easiest to write. I often have people start with this when I am writing a full paper all at once. We've broken our papers up into sections so that you're not going to write one full paper. You're just going to hand in each section at a time. So what are the exact instructions you followed to get to the result? Someone should be able to read them, replicate your experiment exactly. This is typically written with subsections. So this is APA format here. So your level 1 heading should be centered, bolded, and that would be your method section. So each of your section headers should be a level 1 heading. And then subsection headers would be level 2 headings. So you can see here in this example, I have grouped this into fermentation ability tests and large extracellular molecule use. So these are two different types of tests that are all similar. And then within that, if I change my color here, I have two different tests that test for similar things, but they're going to have different methods, right? So at the red level that we have here, you have a subsection header that's a level 2 heading. And same thing in the bottom red level here, you're going to have a subsection level 2 header. So this would be left aligned, bold, and then you'd move on to your next section header. So your subsection might be fermentation ability tests. You might not group them like this. You might just label each test, and then each test would be a level 2 header. You don't have to have 3 levels. So you might have phenol red test as this top level. Just know that the further level you go down, there is a different structure for each. So level 1 should be centered and bold. Level 2 should be left aligned and bold. Level 3 should be left aligned, bold, and italicized. And then your text is just regular font, left aligned. So that is APA format for section headers, which is going to be the most important part of your methods. I think that's the thing that people stumble with the most here. The results. The results gets tricky for people. So this is your what. What did you find? This is only a statement of the data in figures and tables to help see that. You're not going to interpret it here. So you're not going to relate it back to your introduction of why it's important. You would tell us what it is. So like, I observed bubbles in my catalase test, and therefore it is catalase positive. That's the type of interpretation you would do, not I observed bubbles, therefore it's catalase positive, and this is important because it's found in an area with lots of reactive oxygen species. That last bit would be part of your discussion. So the results, you're just telling us what you found. So you are going to use all of your figures and tables in this section to help represent that data. You have to write it in paragraph form saying this test resulted in blah, 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 blah. So you're going to have all this data that's just kind of you're accumulating this whole time. Hopefully in your lab manual, it looks more like this, that you've sorted it and have some understanding of what it looks like. As you're preparing your paper, you're going to arrange it and visually present it. So you've got these sort of two next steps of what you're doing with your data. So these are going to be your graphs, your tables, your figures, your pictures of your plates, your pictures of your tests and colors and things like that, so that you can explain what you found. The explanation with a story, that's for your discussion, not for your results section. So your discussion is where you're going to say, hey, all of these red blocks formed the door, sort of thing. This is what led us to our discovery of our unknown microbe. This is where you sort of explain it to everyone. But the results section is where you're just going to go step by step and tell us what you found and where you came to your conclusions. All right. That leads us to discussion. It's your explanation point. So you're going to discuss any errors, difficulties you encountered. You're going to compare your results to published research. Do your results follow what's already been found, or is it different? And what would you do differently or do next as a result from this experiment? So this is where you really get to talk about what's going on. Think about this like a conversation with your reader about like why this was cool, why this was important. So this should be a longer section and should have references in it. Again, references, don't plagiarize. Putting it in quotes is not enough. So for our class, we are going to require that you have a certain number of primary sources for each different section. So a primary source is the reporting of an experiment, study, or any other new research. So if you had gone and interviewed someone, that would be considered a primary source because you physically interviewed them and this is a production of new information. For us, we're focused on science. So when you do an experiment and you tell the scientific community, I did this thing and found this, that's a new research, new information. So a key here is that these papers typically have a methods and a results section. So if you're looking and you're having a hard time trying to decide if your paper is a primary or a secondary source, check really quickly to see if it has a methods or results section. That's going to key in and tell you that it's probably a primary source. Secondary sources summarize other primary sources. So they typically have the word review or summary in the title. Their section headers, instead of being abstract, introduction, methods, results, will be really descriptive. They'll be like mechanisms of action in the cell, mechanisms of sugar fermentation in blah, blah, blah. And they're going to continue to have really big, long titles rather than introduction, methods, results. So those are secondary sources. You can think of those as papers that really tell you lots of information. And these are great. They're still reputable sources. These are still published in journals. They're just not primary sources. So we want you to have new research and summaries when you are reporting things for our papers. So news articles are still considered reputable sources, but they're not primary sources. They are secondary, but they're not secondary research articles. If a news article is talking about scientific findings, you must cite the original journal article that the findings were reported in, not just that news article. So if you found something in the New York Times that was like, cool, we found this microbe that's producing new antibiotics, that's awesome. They're going to tell you which researcher was doing it and where it was published. Then you need to go back and find that article in the journal and cite that for your paper. We typically won't use news articles in scientific writing. It's just not considered rigorous enough. So you need peer-reviewed, and peer-reviewed can be primary or secondary. You can pretty much assume if it's in a journal that it's peer-reviewed. If it's a news article or public source, public information, it's probably not rigorous enough for our papers. All right, the other one that tricks people up, is the methods of study. So remember, primary sources typically have a methods and results section. The one that gets people most often is systematic review or meta-analysis. This is typically a primary source because they review a bunch of other research, they analyze that data, and they come up with new conclusions. So they are producing new information. The way you will know that it's a review article versus a systematic review is that a systematic review will have those sections. They'll have a methods and results section. If the title just has review in it, then it may be a review, a secondary source, rather than a systematic review. These are all ways to set up your study. So you have your traditional experiment, your control with an independent and dependent variable, and you test a hypothesis. It's very direct, point A to point B. Then you have observational studies where they just observe things happening in the natural environment. You don't do an outright test, you're just reporting on what you found. Case studies are used most often in medicine, so they're interpreting individual historical cases to get a better understanding of a topic. So this is typically a patient presented with something interesting and different that we haven't seen yet, and we are telling the world about it and how we handled it so that we can better understand it. Correlation studies are going to measure relationships between variables because there's too many variables to be able to control them. And then there are many, 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 many more methods of study. There are more experimental design setups than these. These are going to be your most common that we run into for our experiments. And what we're worried about is really this last one often confuses people. So remember, primary sources have a methods and a results section. Secondary sources typically have descriptive topics as their section headers. Sometimes they will use the word review in the title. Common science writing issues. I really love this comic because I think everybody can relate to being called by their full name by their mother when they're in trouble, as can apparently E. coli. So, formal tone is our biggest issue. I'm going to skip that for a minute and just talk about the next topic because that's where our comic is. So you want to make sure that the first time you mention a bacteria, you need to write their full name before you can abbreviate it. It's just like using any other abbreviation. If I were to write a topic about the American Red Cross, I could say American Red Cross, and then you'd put in parentheses ARC would be my abbreviation. So you'd continue talking, and later you could then say the ARC provides first aid and CPR training as well as babysitting. But I couldn't just start with the ARC provides those. You need to tell us what the ARC is, American Red Cross. So it's the same thing with bacteria. You need to tell us what the full name is, Escherichia coli, rather than just E. coli. And then you can abbreviate it after that. You don't need to put it in parentheses like a typical abbreviation after that first one. It's just implied as you keep going. When you're writing these down, your genus and species get italicized. The genus gets capitalized, the species does not. So that is the one thing about this comic that is wrong. Coli should be lowercase, all of it. So genus and species both get italicized when you're writing it. Genus is capital, species is all lowercase. Okay, back to that formal tone. So when we're writing for science, you kind of want to sound almost stuck up. You want to be very pompous. You need it to be very formal. No, I did this, we did that. That's the thing we saw. You want to make sure that your sentences are short and concise, and think about it like you are writing a formal letter for a job. You are writing a formal letter for science telling us what you found. So no colloquial terms, no like slang or written language. You really want to make sure that this is an elevated paper. If you have trouble with this, we can talk about this some more and give you some help on making your tone more formal and concise. Table and figure formatting is one that gets everyone. Table and figure formatting needs to be in APA format. We go back to that heading. We talked about the subheading levels. Tables and figures get formatted in a specific way. They have what's called a figure legend or a table legend. So when you're reading a paper, as you do your first worksheet, read your papers and see where they put in a picture or a graph or any of those things. How did they introduce that? How did they reference it in the writing? So you're going to have written your sentence and say, a summary of all tests are available in table 1 or represented in table 1. And then table 1 is labeled, but maybe I have 2 tables. So the next one I talk about would be labeled table 2. There is a proper way to format that. The table 1 or figure 1 or table 2 or whatever order they're coming in should be bolded. It's just the word table and the numeral 1. There's no period, there's no comma, there's no colon or semicolon after it or dash. It's just table 1. Then on the next line down, it would be the title of your table. So figure 1 or table 1 is in bold on its own line. The next line down is the title. That should be italicized on its own line. So figure 1, then the next line down is title, and then your image. And underneath that, you'd have a short description telling us anything specific or of note in that. Purdue's online writing lab has a great APA format resource. It's linked on our Brightspace. I suggest you use it. I still reference it pretty frequently. I think I've already hammered this home. You cannot quote directly from a source. You need to paraphrase it in your own words. It's a skill you need in science. And then everybody mixes up whether it's a primary or secondary research article. So a primary research article has a methods and a results section. A secondary research article is a summary or a review of other people's work. Primary produces new information, secondary summarizes it. All right, and then tools. Zotero, please, if you've never used it, download the program. Zotero is a citation management program. So it will add in a widget on your internet browser, and you basically, whatever paper you're looking at, you just click the button and it saves it to Zotero for you. Then when you're working in your Word document to write your paper, it will have an add-on for Zotero, and you just click add citation from Zotero, and you can just click on it and it will automatically cite it for you in APA format. You do not have to memorize how to do APA format for every single one. If you can't get a hold of a research article, Interlibrary Loan is a wonderful tool. They'll typically get it to you in about 24 hours. You do need to register for an account with Iliad. It is not just your regular school account. You'll have to register for an additional one with that. Google Scholar, you can typically get most PDFs or most research papers directly from Google Scholar. This is where I start most days. If I can't find an article from Google Scholar that we have access to the whole thing, then I go to our library page and search for it there to see if we have access through our institution. But Google Scholar does a pretty good job of making your life a little bit easier. And then please, please, please explore the resources tab on Brightspace. There's so much more that I put on there for you to explore. So as we're going forward, you're going to work tonight or over the next couple of days in this first assignment on your searching sources worksheet, searching and interpreting sources. Then this weekend, you're going to write your abstract for your microbes in the environment assignment. Next week, we're going to start our unknown microbe unit, and we're going to continue building these sections. So you're going to write an introduction, then you're going to write the methods and results, and finally you'll write the discussion. I don't want you to group this all into one big paper, but we're going to go one by one and focus on each of these sections and build our skills in writing a scientific paper. If you have any questions, please feel free to email me, natalie.barrett@maine.edu. I am looking forward to reading your papers.